Welcome back,
Hello,
everybody.
everyone. Hi, welcome back, everybody. This is Tal Zaks here. Again, it's a real pleasure for me to kick off our section on prophylactic vaccines, infectious disease vaccines. This is an area where clearly I think there's been a lot of focus around COVID, but importantly, we have been advancing our lead vaccine here, Cmv, and have promised you some news as it regards to our ability to dose select, and we're happy to share with you today the interim data from the phase 2 that points to the continued progress of this, and it'll be followed by the progress in other of our vaccines, and at the end we'll come back and talk about COVID as well. With that, let me hand over the microphone to Lori Panther, who leads our CMV program. Lori?
Thanks very much, Tal, and good morning, everyone. I'm Lori Panther, Senior Director of Clinical Development at Moderna, and I'm the medical lead for our CMV program, and it's been pretty busy and exciting for the CMV team lately. Now, developing a CMV vaccine is a journey that not many have attempted, and at the end of our journey, we want to be telling the world that we're improving the health of our kids, preventing birth defects, preventing hearing loss, and giving these kids the opportunity to have a normal life. So in these next minutes, I'm going to be updating you on our progress toward that goal. Next slide, please. We now have 2 trials ongoing, and we're preparing for phase 3. I'll be talking this morning about the results of a recent interim analysis from our phase 1 trial, and in addition, I'm very excited to be sharing with you the first interim analysis results from our phase 2 trial. Next slide, please. Now, cytomegalovirus is a pretty common infection. I'll bet about 2/3 of the people on this call have been infected with CMV, and I'll bet almost all of us don't know it because it never made us sick. So why would we pursue a vaccine against it? Well, it's all in the timing of when that infection occurs. If a pregnant mom has CMV infection, she can transmit it to her developing baby, and it can be at a crucial time when that baby's brain, vision, and hearing are developing. About 3 out of 10 moms who are infected with CMV during pregnancy will have a child with congenital CMV infection. And about 10 to 15% of those kids will be extremely sick in the labor and delivery room. Some won't even survive to see their first birthday. And those who do survive, the majority of these sick babies, will invariably struggle with lifelong physical and developmental disabilities. But even kids with congenital CMV infection who look fine in labor and delivery can be at risk for developing hearing loss and cognitive delays in childhood. Our team thinks of these kids every day. And to have a safe and effective vaccine and to prevent some of this hardship is just a huge opportunity for us. Next slide. So a bit of a reminder about our CMV vaccine. It contains 6 mRNAs, which encode for 2 crucial CMV antigens. One of these 6 mRNAs encode the Gb antigen, and this is a protein on the virus that helps it infect many different cell types in our body, including cells called fibroblasts. The other 5 mRNAs accomplish an amazing task because they encode 5 proteins that make up this complex antigen on the virus called the pentamer. Now, pentamer is important because it's crucial for entry into our epithelial cells, and it's really good at stimulating the human immune response against the virus. But it's also really, really difficult to make outside the cell. It needs the cell to process it and to fold it just so in order for it to function. It's hard to whip it up in a lab and give it as a vaccine in that way. Really, the best way to make it is to have our own cells make it, which is exactly what mRNA vaccines do. Lastly, it's important to note that the only CMV vaccine to complete an efficacy trial has been a vaccine that has included only the Gb antigen. And those trials showed a vaccine efficacy of around 50%, and 50% is not bad. But the problem was that the durability of the immune response was not optimal. So with the addition of pentamer antigen to the Gb antigen, we're expecting a better immune response that translates into effective protection against CMV infection. Next slide. So here's the news on the status of the ongoing trials. For the phase 1 trial, last month we reached our 12-month analysis, which gave us some early indication of the persistence of the immune response. And for the phase 2 trial, we began enrollment shortly after the first of the year, and it went quite quickly. We were fully enrolled by early March, and we've now reached our 3-month interim analysis of safety and immunogenicity through those first two vaccinations at all of the dose levels. Now, it's important to note that our challenge here was that the second vaccinations were scheduled at about the same time that the COVID pandemic was hitting the U.S., and the team quickly made adjustments so we could complete those second vaccinations, but also keep our participants and our study personnel safe. Bottom line is that we were able to include approximately 65% of the total study enrollment for this 3-month interim analysis. Next slide. Now, note that the overall aim of our phase 2 trial is to prepare for our phase 3 trial. We aimed for this phase 2 trial to provide the information we need to choose a final dose for that phase 3 trial. And with the vaccine preparation that we'll be using in the phase 3 trial. There were some changes implemented to get from the phase 1 to the phase 2 presentation, namely the ratio of the 6 mRNAs in the vaccine were optimized for improved immunogenicity. The manufacturing process was optimized to improve tolerability of the vaccine. And the vaccine's not in liquid form anymore. It's lyophilized for implementation in phase 3 and beyond. All of that said, our phase 1 trial continues to teach us about the performance of this vaccine. Next slide. So, some phase 1 data. As I mentioned, in August, we reached our 12-month interim analysis for this trial. And this trial is testing dose levels of 30, 90, 180, and 300 micrograms. The safety outputs revealed no surprises over all 4 dose levels, and there were no serious adverse events related to the vaccine. It was well tolerated and noted that the 2 highest dose levels, 180 and 300 micrograms, had similar safety profiles. The exciting news is the immunogenicity data 6 months out from that third vaccination in the 30, 90, and 180 microgram dose levels. And as a reminder, because I'll be using these terms a lot, neutralizing antibodies against epithelial cell infection is the measure we use to look at immune response to our pentamer antigen. And neutralizing antibodies against fibroblast infection is a measure we use to look at the immune response to our Gb antigen. What we saw was in our seronegative folks at the 90 and 180 microgram dose levels, at 6 months after that third vaccination, the titers against our pentamer were very encouraging, with GMTs 3.6-fold and 3.9-fold higher than the benchmark of a naturally infected seropositive population. And the titers to our Gb antigen remained around the natural infection benchmark titer. And in the seropositive group, the immune boost to both pentamer and Gb remained pretty robust, with geometric mean ratios between 14 to 31-fold for the immune response to our pentamer and 6 to 8-fold for the immune response to our Gb antigen. So the summary here is that our phase 1 trial recently gave us pretty encouraging news showing early immune persistence to this vaccine, just as our phase 2 trial was taking off. Next slide, please. So, on to our phase 2 trial. This trial has enrolled 252 participants, both seropositive and seronegative. The dose levels being tested are 50, 100, and 150 micrograms. And this range was chosen after detailed analysis and modeling of what we might expect of this lyophilized presentation with all of the optimizations I mentioned. And also, as I mentioned a few slides back, the COVID pandemic has been a challenge for all of us, and this trial was no exception. And we amended the protocol to widen the second vaccination window so these vaccinations could happen safely for everyone involved. And as a result, we were able to include 65% of the total enrolled for this three-month interim analysis. First, the safety data. This table shows the frequency of solicited adverse reactions after the first vaccination for both the seronegative and seropositive groups. And as a reminder, when we collect solicited adverse reactions, is when we have each participant fill out a symptom diary every day for the first 7 days after each vaccination. And the symptoms we ask about are pretty much the same across all vaccine trials. They're listed on the left-hand side of the table. They're divided into local adverse reactions, so any symptoms in the arm that was vaccinated, and systemic adverse reactions, so any more general whole body type symptoms. And what we saw was that the most common local adverse reaction was injection site pain, and the most common systemic adverse reactions were headache, fatigue, and myalgia. And this is essentially the same profile we saw in our phase 1 trial. And also important to point out, there were no serious adverse events reported, and there were no participants who discontinued the trial due to an unsolicited adverse event. Next slide, please. This next table has the same layout and shows the frequency of solicited adverse reactions after the second vaccination for both seronegative and seropositive groups. Here, we're seeing the same profile for the most frequently reported adverse reactions that we did after the first vaccination. And two things to note. Number one, there wasn't a distinction between dose level and adverse reaction rates. And number two, there wasn't a notable difference between the adverse reaction rates in the seronegative groups and the adverse reaction rates in the seropositive groups. So, and also of note, and this isn't shown on the slides here, is that fever was much less frequent in this phase 2 vaccine product compared to the phase 1 product. And in general, the observed reported frequencies of systemic solicited adverse reactions are lower in the phase 2 trial compared to the phase 1 trial thus far. So these are gratifying observations because one of our aims with the modifications to the phase 2 vaccine product was to do just that, to improve tolerability. Next slide. Moving on to immunogenicity, this table shows immune response to the vaccine in our seronegative group. The top half of the table shows the immune response to our pentamer antigen, and after the second vaccination, we're seeing GMTs between 49,000 to 57,000 across those 3 dose levels. In the blue circles, we're comparing these titers as a ratio to the baseline titer of our seropositive group. Now, this seropositive benchmark is kind of the goal we set for our seronegatives. It represents the antibody level of people who have had CMV infection in the past, and this is important. What we know about pregnant women who have had CMV infection in the past is that they have about a 10 times lower chance of passing CMV on to their babies. So the hypothetical question we can pose here is, could our vaccine give a seronegative person the protection of a seropositive person? And the answer is, again, in the blue circles, yes, by at least 12-fold with this 3-month IA data. The bottom half of this table shows the response to our Gb antigen, and after the second vaccination, so the 3-month time point, GMTs generally hit at or a bit above the seropositive benchmark over all 3 dose levels. So this is very encouraging news. Next slide. This table shows our results for our seropositive group. Same layout, except that the comparison here is against each group's own baseline titers since they're seropositive to start with. And what we can see here is a robust boosting response across all dose levels, with GMRs ranging 20 to 32 for response to the pentamer antigen in the blue circles, and GMRs of at least 2.5 for response to our Gb antigen. So also very encouraging. Next slide. These next 2 slides are graphic depictions of the immune response tables you just saw. This one shows the progression of immune response to the pentamer antigen, so neutralizing antibodies against epithelial cells. The dotted lines represent the seropositive group, the solid lines represent the seronegative group, and that black horizontal line is our seropositive benchmark value. And so also note that the y-axis is a logarithmic axis. The main focus here are the data points at the far right of the figure. We see a clustering of the seropositive and seronegative GMTs at month 3, so 1 month after that second vaccination. So the immune response in the seronegative folks was similar to the boosting response in the seropositive group. And again, focusing on the solid lines, seronegative dose levels, we don't see a distinction between dose level and immune response at that 3-month time point. Next slide. And this one shows the progression of neutralizing antibodies against fibroblast infection, which is our immune response to our GB antigen, and same layout here. And again, the far right side, at the far right side of the figure, focusing on the solid lines, seronegative groups, we're not seeing a dose level distinction, and we are seeing these GMTs cluster around our seropositive benchmark level. So very encouraging observations that we're seeing. So these phase 2 interim results were key data that informed our choice to take the 100 microgram dose, next slide please, into our phase 3 trial. And to further prepare for the phase 3, we're amending the phase 2 protocol to add more participants to achieve a robust safety data package at that 100 microgram dose level. We're planning our phase 3 to begin next year, 2021, with a primary endpoint of vaccine efficacy against primary CMV infection in women aged 16 to 40. And we're planning to carry this out in a study population of less than 8,000 participants. We sought and received very constructive feedback from a joint national scientific advice meeting earlier this year, and we're deep into operational preparations for this pivotal trial. Next slide, please. Lastly, we're preparing for the launch of our vaccine in a broader sense. We've been focusing on and supporting efforts to really amplify public awareness of CMV and its impact on the health of our kids. There's certainly work to do here, and we have a tremendous team at Moderna focused on this effort. So in summary, the CMV update here is that there's a lot of exciting news. Our phase 1 trial is showing some early indication of immune persistence. Our phase 2 trial is telling us that our intended phase 3 vaccine product is well tolerated and immunogenic, and we've been able to select a dose going forward. And our phase 3 trial preparations are keeping us very, very busy. So thanks for your attention this morning, and I'll hand the screen over to Allison.
Thank you, Dr. Panther. If we could move to the next set of slides, the pediatric respiratory vaccine slides. Good morning, good afternoon, everyone. I'm Allison August, and I'm a Senior Director of Clinical Development in the Infectious Diseases Division. I'm the medical lead responsible for the pediatric respiratory vaccine programs, and I'll share a brief update with you on the status of these programs. There are several pathogens that contribute disproportionately to the burden of respiratory illness in young children. And this burden is dominated by 3 pathogens: respiratory syncytial virus, human metapneumovirus, and parainfluenza virus type 3. We are very excited to be pioneering mRNA vaccines in children through the development of vaccines targeted against these 3 dominant pathogens to address the persistent respiratory disease burden in this pediatric population. I'll now focus on a more detailed update of our ongoing pediatric programs, and I'll discuss a future vaccine option which leverages one of the strengths of our platform, the potential for a combination pediatric respiratory vaccine. Next slide, please. Why RSV? RSV is the largest cause globally of severe lower respiratory tract infection in young children. This has been recognized for decades, and yet it remains unaddressed by a vaccine. Most children are infected at least once by the age of 2 years at an estimated annual cost of $2 billion in the United States alone. The acute clinical presentation commonly leads to hospitalization due to severe bronchiolitis or pneumonia, and there's also evidence of an association between early childhood infection and the development of chronic sequelae such as asthma. Next slide, please. This slide illustrates the mRNA 1345 RSV vaccine, which is engineered for an optimal immunogenic response. Specifically, the mRNA encodes for the fusion protein with modifications that enable the protein to be stable in the pre-fusion conformation, which is critical as it then displays the key epitopes that have already been shown to be superior antigens. Additionally, the pre-fusion F protein is anchored to the membrane, which subunit vaccines cannot do, and the formulation uses the same LNP as several of our other vaccines. Next slide, please. This slide illustrates a high-level overview of our ongoing RSV vaccine study, which is actively screening adult participants. As with our first pediatric study, our Hmpd Pid study, the design for this pediatric study was carefully vetted with pediatric infectious disease experts, pediatric vaccine investigators, and the FDA. Many of the design elements here are standard. We'll evaluate safety and immunogenicity, with the primary endpoint being safety. As such, this study will be conducted with the oversight of both an internal safety team and an external independent Dsmb. The members of our Dsmb, or Data Safety Monitoring Board, are all very well seasoned pediatric vaccinologists. As outlined on the right side of this slide, this study has a small adult lead-in cohort. We then immediately age de-escalate to the target pediatric population of children 12 to 36 months of age. Again, while the paramount goal for this pediatric study is safety, a reminder that we are already dosing children. Next slide, please. Our second pediatric vaccine that's under development will address the two other major causes of respiratory illness in children, human metapneumovirus and parainfluenza virus type 3. These pathogens account for a near equivalent burden of severe respiratory illness in young children that is similarly unaddressed by a vaccine. The clinical presentation with these infections can range from mild symptoms to, again, severe lower respiratory tract illness, including bronchiolitis and pneumonia requiring hospitalization. Of note, there have been no prior attempts to address both of these pathogens in a combination vaccine. Next slide, please. This slide illustrates our mRNA 1653 Hmpv PIV vaccine. It's notably distinct from the RSV vaccine that we just reviewed in that this is a combination vaccine. It targets two diseases simultaneously. This vaccine highlights an aspect of our technology that we believe differentiates our messenger RNA approach, the ability to make combination vaccines. As detailed in the upper left-hand corner, this vaccine contains two mRNAs, each encoding an antigen from a different virus, though formulated within a single lipid nanoparticle. It's similar to the RSV vaccine in that all three contain mRNAs that encode for the fusion protein. All are anchored to the membrane, and all are formulated using the same LNP. Next slide. This slide illustrates a high-level overview of the ongoing Hmpd PIV trial. This study, similar to the RSV vaccine study, has an adult lead-in cohort, which has completed enrollment. We've also completed enrollment of the first 10 pediatric participants in cohort 1, and then had a study pause due to disruptions related to the COVID-19 pandemic. We used the pause as an opportunity for an ad hoc safety monitoring committee meeting to review a preliminary data set on this small initial group of pediatric participants. The SEC, again composed of pediatric vaccine experts, recommended continuation of the study with no modifications in the planned trial execution. This study is resumed. We're actively screening pediatric participants, and we look forward to updating you on our progress and the corresponding full data. Next slide, please. So what's on the horizon for our pediatric vaccine programs? As just demonstrated by the example of our Hmpd PIV3 vaccine program and identified earlier by Stefan this morning, we believe our platform is uniquely well positioned to enable us to formulate combination vaccines. Given the overlapping epidemiology of infection of these three pathogens, three major pediatric respiratory pathogens, a combination vaccine would provide a substantial clinical advantage. The challenge for us is not in the scientific validity or the feasibility to execute clinically or even in the CMC, rather in the optimal clinical development path to a combination vaccine. We're designing a trial to integrate the endpoints against these three different viruses into one development path. Simultaneously, we continue to progress both the RSV and the Hmpd PIV vaccines independently, and likely through phase 2 in dose selection prior to combining them. Next slide, please. Our ultimate goal is a combination pediatric respiratory vaccine. There are clear advantages to the combination vaccine for sure. It would enable a simpler pediatric immunization schedule, happier kids, happier parents, as well as help to the primary healthcare providers. A combination vaccine will also increase compliance and the overall uptake of and public health benefit from the vaccine. It's now my pleasure to introduce Professor Terry Nolan from the University of Melbourne, who will share his insights on pediatric vaccine development.
Hello everyone at Moderna. This is Terry Nolan from the Peter Doherty Institute for Infection and Immunity at the University of Melbourne. So pleased to be able to speak to you today, and sorry I can't be there with you. Unfortunately, the time zone's the big killer, and we have to make do with this. I'm delighted to have been asked today to give you some insights and perhaps a few thought-provoking observations on vaccines, particularly with respect to the prevention of children's diseases. And I've titled this brief talk, Epochs and Opportunities, because I'm going to focus on some of the achievements that we've seen in the last 20 or 30 years in particular, and what I believe to have been responsible for those, a sort of a helicopter view, and following that, some opportunities that are particularly relevant to mRNA vaccines. The advances that we've seen in disease control have been spectacular since the 1950s, with polio, with diphtheria, tetanus and pertussis, with the many different organisms that can cause meningitis. I'm thinking of Hib as well as pneumococcal disease and also meningococcal diseases. Diarrheal disease with the advent of an effective rotavirus vaccine, and then pneumonia with conjugate vaccines, in particular conjugate pneumococcal vaccines, made a massive difference to invasive pneumococcal disease and also to pneumonia, and to other conditions which we rarely see these days in those countries who have high rates of vaccine uptake: measles, mumps, rubella, and now varicella. I'm going to suggest there are three basic streams or reasons which have contributed very substantially to these epochs, with quantum shifts in disease control coming close to elimination for some, such as polio, of course, and for others, very close to very high levels of disease control, such as meningococcal disease, for example, Hib, and many others. So the first of these is advances in vaccine science or vaccinology. Broadly speaking, especially since the 1990s, the advent of conjugate bacterial vaccines, which in the beginning were simple, such as with Hib, for example, but then with more complex multiple serotype vaccines, the conjugate pneumococcal vaccines have been wonderful examples of new technology that was previously not available. Reverse vaccinology was an important new step that resulted in the first effective meningococcal B vaccine, a completely different approach to understanding the genome of the organism and its protein structure, which in turn allowed rational vaccine design based on identifying and targeting specific areas of the bacteria which were important for prevention of disease. And then finally now, the era of nucleic acid vaccines, I think it's fair to say, has arrived. And we're seeing that with Moderna's input in particular into the equation after many years of DNA vaccines struggling. This has been a wonderful new addition to that epoch. The second broad thing that's happened almost unnoticeably is the advent of multiple combination vaccines. Of course, combinations were available in the 50s with DTP vaccine, but and then later with measles, mumps, rubella, and so forth. But it was really the large infant combination vaccines in the late 90s, early 2000s, which has really made a huge difference to accommodating the schedule, to allow new vaccines to be added, and to deal with issues of concern about doing too much all at once to children, something I'll come back to shortly. Other combinations, more recently MMRV and meningococcal combinations especially, some of which have come and gone as more effective combinations have appeared. I'm thinking of Hib-Men-C, Hib-Men-CY, quadrivalent meningococcal vaccine, ACYW, and also in trials now, and not far away, a 5-valent or 5 serogroup vaccine against meningococcus A, B, C, W, and Y. But at the same time, there's been, if you like, shifts in society which, despite the fact there have been very significant noise attached to those who are fiercely opposed to vaccination, fortunately in very, very small numbers, even in the US, certainly elsewhere in the world, and more generally a concern about being absolutely assured that vaccines are safe, which is now described as vaccine hesitancy or vaccine confidence. But there has been a shifting tolerance, I've noticed, in governments, certainly my own government in Australia, both federal and state governments, shifting tolerance of freedom not to vaccinate. And this has resulted in changes in, if you like, exceptions to mandates, which mean that either religious objections or in some cases personal conscientious objections to vaccination are no longer permitted. And this has come not at the behest often of those like me, people who are involved in vaccines and promoting them, but actually has come from the politicians. And I've been surprised at how receptive they've been to the need to promote vaccination. Sometimes worried that they've gone too far, but nonetheless, it has been a shift. There is now a view that herd immunity is everyone's responsibility, and that tolerating 5 or 10% of the population as not needing to be immune because the herd will protect them is not seen any longer as a reasonable reason for them not to be vaccinated. So this notion of combination vaccines, I want to just explore a little bit more. They have made the infant schedule feasible as new vaccines have emerged, as I mentioned, and the potential for further efficiency does exist and probably hasn't been fully realised. We've just done a review published earlier this year of the first 10 years of experience in disease control terms of a hexavalent infant vaccine, which is the anchor point of the Australian schedule. This is a GSK product, but there are other multivalent 5 and 6-valent vaccines used around the world now, and it has become the anchor point of schedules everywhere. But we were interested in just seeing what happened with disease control as this became that anchor point. And clearly, the impact on disease control has been massively successful. It's been mediated through very high levels of uptake of the vaccine. Obviously, having the combination in and of itself alone is not the reason, but it's permitted the promotion of vaccination in a crowded schedule to actually achieve this wonderful outcome. And the reason why combination vaccines have been successful is obvious. It increases coverage across half a dozen conditions all at once, rather than having to schedule a number of different appointments or a number of different time points in the schedule. And so if you like, those that for which there may not be so much enthusiasm get caught up in the overall improved coverage. There's convenience for parents and also for healthcare providers. The cost of materials is reduced, storage is simpler, marketing is also simpler, rather than worrying about exactly what's in the combination vaccine. Generally speaking, the combination itself becomes known as the children's vaccine and worth getting. And finally, in terms of parent acceptability, this notion of worrying about a pincushion or antigen overload and anxiety, I think paradoxically has been helped by having combination vaccines, because the numbers of actual separate injections or separate vaccinations is what parents often see and are concerned about, as was shown in this national survey done in the US by Bruce Gellin and others. Kathy Edwards was one of the authors of this paper, a national telephone survey. I just wanted to point out on the bottom line of this chart that 25% of parents who were surveyed here, these are parents of children aged 6 years and under, the 25% felt that they were concerned, had serious concerns about their children's immune system possibly being weakened by too many immunisations. Even at that time, of course, the view may be different now. I'm not sure that this particular survey has been repeated more recently, but the concern that doing more and more with the children's immune system is something which we as promoters of vaccines need to recognise that and explain and be part of the discourse in promoting vaccination. I want to switch now just very briefly to thinking about the future and what the next epoch might look like, and in particular the identification of respiratory viruses as high-value targets. I believe elsewhere in the program you'll see some of the epidemiology which underlies the basis for, if you like, what's called often the unmet medical need, which is demonstrable. But in summary, there's clearly a continuing case and need for better influenza vaccines. We've got good vaccines, but they're not good enough. The fact that they need to be renewed annually is an issue, and until there is a technology which is going to overcome that, we're stuck with it. But in particular, the effectiveness of those vaccines which are given annually is not where we'd like it to be. It's in the 50 to 60% range at best, rather than the 90% plus range where we'd like to see it. Secondly, respiratory syncytial virus, or RSV. There's been a more than 50-year quest now to identify an effective vaccine against this nasty virus, which affects particularly very young infants with high rates of hospitalisation. Potential links to future development of asthma, which until we have a vaccine we won't really know for sure. But that quest for the vaccine has been harmed considerably by early adverse effects in the late 60s, early 70s of a vaccine candidate with associated enhanced respiratory disease that led to death in some infant subjects because of enhanced infection once they were exposed to the wild virus after having been immunised. There is much better understanding of that phenomenon now and much better technologies which can address that risk. And in fact, there are now many candidates from several platforms, both delivering to pregnant women to protect their infants once they're born, and also to infants themselves. And of course, Moderna is one of those players in this field. Other respiratory viruses which are really important include something called parainfluenza virus type 3, or PIV3, which causes also significant and serious respiratory illness, pneumonia, and it also causes croup. And so an effective vaccine against that is a tantalising prospect and human metapneumovirus, a more recently discovered virus, which can cause significant pneumonia again and increased rates of hospitalisation, another very important target. Is there a niche for mRNA combination vaccines? I think there's some conspicuous advantages that the mRNA construct or platform does offer. The first is the capacity to package more than one targeted disease in a single vaccine. So in effect, a combination vaccine by having more than one type of RNA scripting a particular protein directed at a particular target disease. What the limits for that will be remain to be seen, but in concept, the simplicity of this construct is really tantalising. There's a reduced risk of chemical or immunologic interference between components of a multivalent or a combination vaccine. This has been a problem in the development of those big combinations that I mentioned to you before, and it was overcome, but with considerable difficulty with multiple clinical trials and attempts at changing the underlying construction of those combinations. The other advantage potentially of an mRNA combination vaccine is that it's tunable. In other words, the potential to redesign the vaccine to address antigenic drift, change in the mutation in the virus which the vaccine is directed against, but with a simple alteration in the mRNA script, if you like, without altering the entire construct. This has enormous implications for simplicity in terms of whether further regulatory requirements might exist for further clinical trials or not, and potentially is a huge advantage. And similarly, if it does turn out that mRNA vaccines don't require adjuvants to the same extent that proteins obviously do, this is going to be a huge positive for potential new vaccine development. Sorry about the cough. This slide, regulatory touchpoints, addresses some key issues. I'm not a former regulator, but I've had a lot to do with interacting with regulators over my career. And one of my colleagues, a former regulator, made a simple point about adjuvants, that in general, regulators don't like adjuvants. They add complexity, they add uncertainty, and by and large, it's a much simpler world without them. So obviously there are good reasons for having adjuvants, and they do work and offer considerable advantage in some contexts, but having a situation where they were not required is obviously going to be a big advantage. Secondly, safety. For children's vaccines in particular, there is, well, more generally across the whole age span, this phenomenon is occurring, but especially for children, there is a tangible shifting baseline on risk tolerance by regulators, and increasing requirement for manufacturers and for public health authorities to monitor and provide evidence on safety for vaccines. And as far as the clinical trials that are required, increasing requirements for numbers, for all sorts of other considerations to provide a high level of safety assurance are part of the modern landscape. The assurance of vaccine effectiveness or efficacy, VE, for drifted targets, so again where there have been viruses that have mutated, the regulators these days are much more concerned about monitoring changes in vaccine efficacy over time. This has been very much the case with influenza recently. And if it is going to be the case that there will be important drift in SARS-CoV-2, this will be an issue for future generations of SARS-CoV-2 vaccines and potentially other viruses, including the ones we've just been talking about. And finally, for children, clinical trial complexity is just the nature of the game. Age de-escalation, that is giving vaccines safely to older children before you give them to younger children, is a general requirement for regulatory approval. And with that, either dose de-escalation, reducing the dose as you go down through different younger age groups, also part of that requirement. Another factor which is important, I just want to really, as we come to the end of this talk, feature is the changes in virus ecology from COVID social impacts that have occurred and we've all observed, although possibly haven't observed because people haven't been looking. What I mean by this is this disrupted virus ecology can occur because through the public health measures to contain the pandemic virus, usually with very severe social restriction. I'm speaking from a city which is now getting into nearly 2 months of very severe lockdown, and believe me, we've all had enough of it. But there has been a consequent, as you would expect, impact on virus circulation. That's exactly what it's designed to do, targeted at the COVID virus. But in fact, it also has an impact on all other virus circulation, including influenza, including RSV. I'll show you some data on that in a second, but the reason for mentioning this is that this has a significant potential effect on clinical trials programs, especially for the respiratory viruses we've just been talking about, because some of those programs depend on a proportion of early childhood participants in clinical trials being seropositive, which means that they have had to have been exposed to a virus, usually in the first year of life or so, that most children become positive to RSV by the time they're 2. But if the virus is not circulating, they will not be seroconverting. And in the period when a phase 3 study, an outcome study, will need to be done to look at the reduction in the clinical cases of RSV, for example. This applies to other viruses, but using RSV as an example here. If there is little in the way of circulating disease because of this social restriction, it's going to make doing phase 3 studies harder. It'll take longer, and that's going to be an issue. Here are some data from my hospital, the Royal Children's Hospital in Melbourne. These are not research data, these are practice data. So the axes on these 2 years, the left panel 2019, the right panel 2020, and they show the months from January to August. Remember, our winter period is really from June through August, September. And what it shows is the number of PCRs which were done for presentations. So these are not selected at all. This is all presentations with respiratory illness of children who were given a diagnostic PCR, whether they were admitted to hospital or whether they were just an emergency attendance, irregardless. You'll see there that, first of all, just the red curve on top, the proportion who are positive for RSV, you'll see a massive difference in that curve from 2019 to 2020. And the black bars are the actual numbers of positive RSV identifications that have occurred. And you'll see that in the winter months for us, they virtually disappeared. And what we would have expected was something like what was on the 2019 panel on the left. So there's been a massive reduction in the amount of RSV disease simply because transmission has been suppressed by the public health interventions which have been undertaken. And just to remind you also of where we sit, so to put this in context, on this graph, which shows daily new confirmed cases of COVID-19 from one of the commercial sources of data, the y-axis here is a log axis. So you'll see this is up to date, or relatively up to date, just the last few days. India has now taken over the US, as you can see. These graphs are also color-coded to show the number of, or the test rates within those different countries. But just focusing on Australia on the bottom there, you'll see in these national numbers for Australia are almost entirely in my city in Melbourne. And you'll see we are just now emerging from our second wave, which has occurred during our winter. And we have had really a horrendous time in getting in control of this. It's got into aged care facilities and so forth. And we've gone into this very severe lockdown that I've just mentioned to you. So good luck with your coming winter. I'm hoping that you guys aren't going to have anything like what we've had to live with. You're still up there with what looks to me like still an extended first wave, but I'm surely hoping that you're not going to have to go through what we've just been through, what Spain, Israel and the United Kingdom are about to go through or going through right now. Second last slide, I just want to finish by reflecting on the fact that the objective of all of what we're doing is not to have a licensed vaccine. The objective is to have a vaccine that actually is used in a population and actually does something about reducing disease and death. The way in which we value vaccines is very important. That's what governments really are focused on. And national immunisation programs, or NIPs, where they are publicly funded, there's a way in which that happens in the US is more complex than in some other countries, including Australia, where it's a simpler mechanism, but effectively mechanisms to justify making available vaccines free to the population. The justification for those is based on public funding. The public funding itself is really probably the most important determinant of vaccine confidence in the community and therefore uptake. Of course, it's good to have a vaccine free of charge, but the message that is transmitted to most of the population is that if the government's prepared to pay for this, it must be worth having. And the vast majority of those who go ahead to have their children vaccinated have that underlying belief that the government isn't wasting its time here in investing public resources. It must be worth having. So better methods for cost-effectiveness evaluation is a little concern I have, that we're not yet fully realising the full value of vaccines, and I think we need improved health economic methodologies to do that, understanding the full range of benefits across the life course of a vaccine's impact. Governments are also interested in, obviously, implementability, simplicity in both scheduling and administration, and that's why combination vaccines, as we've discussed, is also very important. But really, in summary, governments are looking for good value for money and high levels of public and healthcare provider demand for vaccines. So, just to finish, 3 simple points that I want to make for today, and I'm hoping that this will help inform your further, more technical discussions on particular vaccines. The first is that the advances in vaccine science that we've seen have provided absolutely enormous quantum shifts in value through primary prevention of childhood diseases, using an amazing array of new science and technology approaches, which is really reaping value for the community. Second is that a much simpler concept, but an elegant solution, is combination vaccines already have a proven track record, and there is significant future potential for their further exploitation. And to wrap all of this together, I do think that mRNA vaccines have a lot to offer in reflecting on all of these opportunities for the coming epoch. Thanks very much. It's been wonderful to have the chance to speak to you today, and I'm hoping that soon many of you will actually be able to come and visit us in Australia, and that I too can come and visit you all in the US. Thanks very much. Good night.
Thank you very much, Dr. Nolan. So my name is Jacqueline Miller, and I joined Moderna very recently to head the Infectious Diseases Development Group. And it's my privilege today to speak to you about our COVID-19 vaccine, mRNA 1273. So on slide 117, you'll see that this vaccine is very special for a number of reasons, but in particular, this is the first phase 3 program that Moderna has embarked upon. Next slide, please. So I don't have to tell anyone on this call about the unprecedented impact that this virus has had on the functioning of day-to-day human life and global economies. This virus was, it's hard to believe, first identified in December, so barely 9 months ago. And since that time, nearly 30 million confirmed cases have occurred worldwide and almost 1 million deaths. In the U.S. alone, we're at 6.6 million confirmed cases and nearly 200,000 deaths. Importantly, this vaccine does not impact everyone equally, and we know that one of the greatest risk factors for severe complications includes increasing age, as well as younger and older patients that have significant comorbidities, so including cardiovascular disease, diabetes, lung disease, and even obesity, so fairly common conditions, particularly in the developed world. So developing a vaccine against this disease has become an urgent global health priority, as currently there's no approved vaccine for COVID-19. Next slide, please. So I'd like to review with you mRNA 1273 and the protein that our single messenger RNA sequence encodes for. So this is, again, a single sequence messenger RNA. It encodes for the spike protein, which for coronavirus is the protein utilized to enter the cytoplasm of the cell. Once inside the cell and with translation, this protein, which has two proline residues substituted, naturally assembles into its trimeric conformation and is presented on the cell surface in the immunodominant pre-fusion conformation. And this is important for two reasons. One, we're presenting the antigen on the cell surface in the same way that it would be presented as if there were a true viral infection, without actually being a viral infection, because there are no viral components in the vaccine. But secondly, that immunodominant conformation is important because what we've learned from convalescent sera is that the greatest neutralizing antibodies are to that pre-fusion conformation. So on slide 120, we show a timeline for the clinical development of this vaccine. And this has been an incredible effort between multiple partners and stakeholders to be able to bring development from initially knowing the sequence in January to launching our phase 3 study in July of 2020, while still maintaining the checks and balances that are necessary as you proceed from one phase of clinical development to the next. And I think importantly, we have managed through close working and reviewing of data through both our partners with BARDA and NIAID, as well as with the U.S. FDA, to review data at critical time points to ensure that taking the next step in development is warranted, while still moving through expeditiously to address this public health crisis. Next slide, please. So on slide 121, I'd like to share with you the study design for the phase 1 clinical trial that has been sponsored by the National Institutes of Health in the U.S. So this was our first time in human study with 1273, which started in March of this year. And the primary objectives were to assess the safety, reactogenicity, and immunogenicity of the vaccine in 3 different age strata: 18 to 55 years of age, 56 to 70 years of age, and over 71 years of age. This was critically important because, as I shared with you earlier, increasing age is the single greatest risk factor for severe complications to COVID-19. So this is a study that was designed to evaluate 3 dose levels: 25 micrograms, 100 micrograms, and 250 micrograms in a dose escalation manner. The data were available in the 18 to 55-year-old cohort prior to moving to the older age cohorts and prior to moving to phase 2 and 3 studies. Importantly, we saw some increased reactogenicity after the second dose with the 250 microgram dose in the 18 to 55-year-olds, and we elected not to continue with that dose level in the older age cohorts. Instead, a dose at 50 micrograms was added. Another important finding from this trial through the 3 dose levels was that the 100 microgram dose was more immunogenic in terms of binding and neutralizing antibodies than the 25 microgram dose. So between the improved immunogenicity profile and the improved safety and reactogenicity profile, the 100 microgram dose has been chosen for further development. And now in the slides that follow, what I will share with you are the results across the 3 age cohorts with that 100 microgram dose. So on slide 122, you see a summary of the safety data we have observed in the phase 1 clinical trial. This is a bar graph in each symptom, starting from top to bottom, the 3 age cohorts, youngest to oldest. We look first at the systemic symptoms as well as at the local injection site symptoms. In gray, you see the reported rates of mild or grade 1 adverse events. In blue, the moderate or grade 2, and in orange, the severe or grade 3. So in the 3 age cohorts after the first dose, the most commonly reported systemic symptom was fatigue, and the most commonly reported injection site symptom was pain. The vast majority of these events were mild in severity. After the second dose, we observed some increased reporting of reactogenicity, and what you will see on the slides that follow is that increased reported rate really corresponds with an increase in antibody responses, and particularly neutralizing antibody responses. The events were mostly mild to moderate in severity, and importantly, they were self-limited in duration. So the majority of these symptoms resolve within 2 days. Some persist as long as 5 days. There were no vaccine-related serious adverse events reported in this trial, and no halting rules for the trial have been met. So as we go to the next slide on 123, we start to see the immunogenicity data across the 3 age strata. And in the slides that follow, you'll see a consistent pattern of data presentation. So moving from left to right, you'll see the 18 to 55-year-olds, then the 56 to 70-year-olds, and then above 71 years of age. This first slide depicts the binding antibody concentrations, and these were measured via an ELISA enzyme-linked immunosorbent assay. Again, subjects were vaccinated with the 100 microgram dose at day 1 and day 29, or 1 month apart. What you see on the x-axis are the antibody concentrations measured through area under the curve at various study day time points, so 2 weeks after dose 1, as well as 1, 2, and 4 weeks after dose 2. And in the pink, you see the middle 2 quartiles of a range of 41 convalescent sera, which were tested with this binding antibody assay. And this is important because we're now able to compare the immunogenicity results from our vaccine with patients that have had natural infection and developed an antibody response. So after the first dose, 100% of subjects seroconvert to the spike protein, even in the oldest age stratum. And these antibody concentrations also exceed the median of those of the convalescent sera. After 2 vaccinations, we see that in all age groups, the antibody concentrations are within the range of the highest quartile of those convalescent sera. Next slide, please. So the previous slide showed you the overall antibodies. These now are antibodies that are actually capable of virus neutralization. So these are neutralizing antibodies that have been measured in a pseudovirus lentivirus reporter assay. And again, you see the 3 age groups moving youngest to oldest from left to right. Again, you see the convalescent sera in pink. So after the second vaccination, the antibody responses were detected in all vaccine participants across age strata, and the levels of neutralizing antibodies were also comparable in all 3 age groups. Importantly, these neutralizing antibody titers are within the upper half of the range of convalescent sera for all 3 age groups. Next slide, please. So on slide 25, we're transitioning from talking about the humoral or antibody responses to the cellular or T cell responses. And in this phase 1 study across the 3 age strata, we investigated Cd4 T cell responses of both a Th1 and Th2 phenotype. So why is this important? There have been some speculations that coronavirus might lead to vaccine-enhanced respiratory disease because of some similarities with previous viruses where vaccines to respiratory syncytial virus and measles virus were shown to induce the Th2 phenotype, which was hypothesized to lead to more severe disease in vaccinees. In our case, what you see are the CD1 or Th1 Cd4 T cell responses in all 3 age groups. And what you can see is that by 1 month post-dose 2, these T cells are induced in all participants. The Th2 phenotype was detected exceedingly rarely. And again, this was reassuring for us to begin a larger scale trial in phase 3 as we begin to monitor for vaccine-enhanced disease. So, in summary from the phase 1 clinical data, the neutralizing antibody titers are observed in 100% of evaluated participants. And this is critically important because we see these antibody responses in the upper half of the range of convalescent sera in all age groups. Again, there was a live neutralization assay that was conducted. I have not shown you those data today, but importantly, they were well correlated with the pseudovirus assay, and that is the assay that we will move forward with in phase 3. And we saw at day 43 geometric mean titer levels at the selected dose of 100 micrograms, also above those seen in the reference convalescent sera. So why are all of these observations important? Well, we observed similar levels of neutralizing antibodies in non-human primates, the rhesus macaque. And the rhesus macaque, while they don't have the same severity of disease observed in humans, they do get the same respiratory symptoms when challenged with SARS-CoV-2. So after these animals are vaccinated and then challenged both in the nose and in the lungs with SARS-CoV-2 virus, the antibodies that are induced are able to protect these animals from infection, even at the lowest doses that we believed would be sub-therapeutic doses. And that's important, again, to reassure us moving into this large-scale phase 3 study about the possibility of this vaccine not only to protect against symptomatic infection, but also to prevent against enhanced disease. So on the next slide, slide 127, I'd now like to start talking to you about the ongoing studies in phase 2 and phase 3. Moderna has sponsored a phase 2 study, which is a safety and immunogenicity study that is investigating 2 dose levels, 50 and 100 micrograms, in 2 different age strata, 18 to less than 55 years of age and greater than 55 years of age. A total of 600 subjects have been enrolled, so 300 subjects in each age stratum. And within each age stratum, 100 subjects in each group were randomized to receive 2 doses 1 month apart of either the 50 or 100 micrograms of 1273 or placebo. We have enrolled healthy males and females above 18 years of age, and we consider these individuals to be historically negative for COVID-19 disease, so they are queried at the start of the trial, but we do not screen subjects out for baseline SARS-CoV-2 serostatus. And with an infection that can be as silent as COVID-19, that's critically important as we begin to assess our vaccine in the field in larger populations. So the study endpoints are going to be safety, and these are going to be similar to what you see in the phase 3 study. We'll look at solicited adverse reactions for 7 days after each vaccination, unsolicited adverse events, which will occur 1 month after each vaccination, and serious adverse events and medically attended adverse events that occur throughout the trial. From an immunogenicity perspective, we will be evaluating binding antibody concentrations with ELISA and neutralizing antibody titers with the pseudovirus assay. The length of follow-up time for all subjects will be 13 months or approximately 1 month after the second dose. So this study is completely enrolled and all subjects have received both doses, so they're in their longer-term follow-up of the study. Next slide, please. So as I mentioned, on July 27th, we launched our pivotal phase 3 efficacy, safety, and immunogenicity study, which will enroll 30,000 participants to receive either 2 doses of 100 micrograms of 1273 or placebo. This study has been branded the COVE study, which stands for the COVID Efficacy and Safety Study, and would not be possible without the partnership of BARDA, who is funding the study, NIAID, who have been significant scientific collaborators for the study, and many other thought partners as we have designed and executed on the study. So unlike the previous 2 studies, this is really the study where we are going to demonstrate that the vaccine will be useful for various populations that need protection against COVID-19 disease. At least 25% and up to 40% of subjects in this study will be at increased risk for severe complications of COVID, either because they're over 65 years of age or because they're under 65 years of age but have one of a variety of comorbid conditions that the CDC has suggested are associated with an increased risk against severe complications. The remaining 60 to 75% of the subjects will be less than 65 years of age and not have a particular comorbid risk. However, they will be living either in areas of the country where infection rates are high or will have an occupation that puts them at increased risk for disease. And importantly, because minority communities have been disproportionately impacted by the burden of COVID-19 disease, we are making enrollment of a population reflective of the U.S. demography a priority for this study. And I'll speak more to that in the slides that follow. Similarly to the Phase 2 study and Phase 1 study, while patients should not have had a history of COVID-19 disease, we are not excluding participants based on their initial serostatus. There are two co-primary objectives of this study. One is to demonstrate the efficacy of the vaccine against symptomatic COVID-19 disease, and the second is to further evaluate the safety and reactogenicity profile. And subjects are going to be followed for approximately 2 years after the second vaccination. And while I'll be giving you some more details in the slide that follow, I'm really pleased to announce that this morning we have posted our full study protocol on the Moderna website. So if you have curiosity after the phone call, you'll be able to look and see some of the methodologies we'll be using. So on the next slide, I'd also like to go over an initiative that I'm particularly proud of, and this is the COVE Study Diversity and Inclusion Advisory Committee. So as I mentioned to you, we are absolutely prioritizing ensuring that the study results that we generate are representative of the U.S. population, and therefore we can have more confidence in the generalizability of those results to the individuals who really need to be vaccinated. The purpose of this committee, which is comprised of various health disparity experts from various groups within the government, are to help us review our enrollment and race and ethnicity demographics on a weekly basis, to help us design outreach activities and outcomes to really engage communities that traditionally have shied away from participating in clinical research. We also want to be sure that we are making it more feasible for subjects to continue the study to completion. And so to that end, they are also supporting us in the development and implementation of retention strategies. Next slide, please. So the study is really well underway, and enrollment is closing in on completion. We've enrolled over 25,000 participants as of today, and the number of subjects that have received their second dose is nearing 10,000 subjects. You can see on the right-hand side of the slide a pie graph depicting the current ethnic diversity, and while it's a little bit difficult to read, we're extremely proud of the fact that we have been able to get to approximately 28% of participation from ethnic minorities and diverse communities. We are now posting this information on a weekly basis to the Moderna website, and the statistics are updated every Friday. So if you have interest to follow after the call, you can continue to follow on a weekly basis. So next slide, please. Now I'd like to transition into explaining a little bit more how we're going to evaluate the efficacy against symptomatic COVID-19 disease and about how the trial will be monitored throughout its duration. So on this slide, we review the case definition for symptomatic disease. And to be considered a case of COVID-19 in this trial, subjects need to have a set of clinical symptoms coupled with a laboratory assay, an RT-PCR nasal swab, nasopharyngeal swab, or saliva swab that's positive for the SARS-CoV-2 virus. These symptoms can either be systemic symptoms, in which case there must be 2 of them, and these symptoms include fever, chills, myalgia, headache, sore throat, and new smell and taste disorders. Or a subject can have a single respiratory sign or symptom, which include cough, shortness of breath, or difficulty breathing, or having clinical or radiographical evidence of pneumonia. The primary analysis is for subjects that are seronegative at baseline, so although we are assessing all comers, and we will certainly be looking at those subjects who are seropositive in some of our exploratory efficacy analyses, the primary endpoint is based on subjects who are initially seronegative. The cases begin to accumulate at 14 days after dose 2, and if you'll recall back to the immunogenicity data that I showed you in phase 1, the reason for that is because we've shown that those neutralizing antibody titers are present at that time. So if we move to the next slide, I'm giving you an overview of how this study is going to be monitored. And I think it's important to emphasize that we have an independent data and safety monitoring board that is looking at this trial. So these are investigators that are part of the committee set up by the NIH, but independent of both NIH and Moderna. And their role really is to look at the clinical data as we progress through the trial in both blinded and unblinded fashion. They're looking for any potential safety signals, as well as the opportunity to tell us that we have met our primary efficacy statistical criteria. So what will happen during the course of this study? We meet frequently with the Dsmb. These are experts in infectious diseases, epidemiology, and bioethics, and they are monitoring both serious adverse events as well as cases of COVID-19 throughout the trial. Each time they meet, they make a recommendation about whether the trial should continue as planned, whether the trial should continue with some modifications, or whether the trial should conclude. And the trial may be also paused if a safety event is observed, but it can also be concluded on the basis of meeting efficacy criteria. Those recommendations are communicated to an oversight group, and that oversight group is comprised of 3 individuals, one from the NIH, one from BARDA, and one from Moderna. So Moderna actually chairs that committee and discusses the recommendations with the oversight group so that we can benefit from the expertise and experience of our partners. Once the oversight group has met, the decision is discussed at senior levels within Moderna. And should we hear at one of our interim analyses that we have met our efficacy criteria, Moderna will then consider the totality of the database, both safety and efficacy, and make a decision about whether to submit to FDA for the emergency use authorization. That review by FDA is also an independent regulatory review, and they themselves also seek independent advice from a committee called VRBPAC, or the Vaccines and Related Biologicals Products Advisory Committee. So the point I would like to underscore is that throughout this process, there are independent reviews of data, both in an ongoing way and once the analyses have been concluded. And that independent review really gives confidence that the data generated in this trial are representative and can be relied upon to give confidence to implementation of a vaccine program. So let's move to the next slide, where I'm going to speak a little bit more about the Dsmb monitoring of the primary efficacy endpoint in a bit more detail. And I'm going to take a couple minutes with this slide because it is a bit complicated. I'd like to start with the graph on the right. So what the graph on the right shows you is the cumulative boundary crossing probability on the y-axis as a function of vaccine efficacy on the x-axis. In any efficacy trial, the likelihood of meeting your endpoints is really based on 3 factors. The overall efficacy of the vaccine. Secondly, the sample size. So larger sample sizes lead to closer refinements of point estimates to the actual efficacy. And then third, the random distribution of events as you go through a trial. So in any given trial, you don't necessarily receive cases that occur in the vaccine group and control group in an alternating fashion. They come into the trial and are reported randomly. So how does this graph help us understand how likely we are at various interim analyses to meet the statistical criteria? Well, if we move to the next slide, what we see is a vaccine efficacy highlighted in blue at 60%. And this is important because 60% is the conservative assumption that we used when we designed the trial. Obviously, we are quite hopeful that the true vaccine efficacy will be higher. And what you see on the left-hand side of the grid is that the first efficacy interim analysis will be performed when 53 cases are accumulated. That's shown in the fine dotted line labeled Interim Analysis 1 on the graph. At Interim Analysis 1, if the vaccine efficacy is 60%, there's a 10% probability that we are able to meet the statistical criteria successfully. But as we capture more cases on the next slide, so now we're talking about the second interim analysis where there are 106 cases accumulated, you see that with the higher sample size, the likelihood of meeting our statistical criteria increases to 65%. And by the time we reach the final analysis on slide 136, at 151 cases accumulated, we have a 90% probability of successfully meeting that statistical criterion. And that's really what we're speaking about when we refer to a study having 90% power. So the study was really designed to look at 151 cases, but because we believe that our vaccine may be more efficacious than 60%, we've designed these interim analyses to allow ourselves the opportunity to investigate the data and potentially conclude the trial earlier based on meeting those criteria. So if we go to the next slide, now we're going to go through the same 3 different analyses, but see what happens when we land at 75% efficacy. So if we move to slide 138, what you can see is with just a 15% increase in efficacy, at the first interim analysis, there's now a 50% probability of successfully meeting our statistical criteria. On the next slide, 139, you see that once you get to then the second interim analysis, or 106 cases of COVID-19 accumulated, the likelihood of meeting statistical criteria exceeds 95%. So I hope that that helps demystify a bit how we will be monitoring the safety and the efficacy of our data while we go through the study. And we really look forward to bringing you more updates of these data as they occur. I'll conclude my presentation, and I'm going to hand over to my colleague Juan Andres, who will speak to you about the manufacturing and distribution of the COVID-19 vaccine.
Thank you, Jackie. Good morning, good afternoon, or good evening. My name is Juan Andres, and I have responsibility for technical development, manufacturing, and quality in Moderna. Slide 141, please. As we discussed in previous meetings, we are a platform, which in manufacturing terms mean that all our products are made in a very similar way. This allows that any learning and improvements that we have had over the years can be applied across our pipeline. Next slide. Because we know the importance of improving our platform, it has been a top company priority to invest in CMMC, chemistry, manufacturing, and controls. The fundamental product understanding has allowed us to make tremendous progress in shelf life, storage temperature, safety and tolerability, potency, and consistency of manufacture. Next slide. Specifically, mRNA-1273, our COVID vaccine candidate, benefits from the progress we have made with all the other vaccines in the pipeline. On the left-hand side, you can see 3 different graphs. Each graph represents a critical quality attribute for the product. Each dot in each graph represents a real GMP batch manufactured during the development of 1273. The values show very high consistency, and you would not be able to differentiate that there are 3 different scales in each graph: small, medium, and large. This level of consistency is what you want to see during the scale-up of a product. Next, please. We presented extensively in March on our manufacturing site in Massachusetts. We wanted it fully integrated, end-to-end, to ensure we mastered all parts of our process. In addition, we designed the plant with a high degree of automation and digital integration to allow rapid growth and scale. Little we knew then that this thinking was going to be essential for scaling up COVID mRNA 1273. Having produced here over 100 GMP batches, in addition to thousands of preclinical and development batches, give us a good and tremendous confidence that we can deliver on our mission to manufacture high quantities of mRNA 1273 COVID vaccine. Next slide. As a reminder, our process is not a traditional biotech monoclonal antibody that requires huge bioreactors. We do not need cells to produce mRNA. No need for product-dedicated plants. Having our manufacturing plant and being an enzymatic cell-free process allows us to scale very fast. Next. Now, we are producing a commercial engine, and this is in addition to the ones we had before, and we will be able to produce hundreds of millions of doses in this infrastructure. Also importantly, this capacity can be used beyond the COVID vaccine for other products that we commercialize after it. We believe this experience is a competitive advantage. Next slide. So, how are we scaling up? Once we decided in our industrial scale, we are replicating units of the same equipment inside our plant and those of our partners. Having the same kit allows for easier replication, faster technical transfer, and a much reduced risk of surprises among different plants. Next. We have designed 2 different supply chains using this concept. One in the US for the US, and another one in Europe for international markets. The magnitude of this effort required us to partner with very reliable companies. So let me expand about them in the next slide. So these companies are very experienced commercial manufacturers, all with extensive experience launching and supplying medicines worldwide. Lonza. Lonza will help Moderna to produce active ingredient, both in the US and in Switzerland. Lonza has an impressive track record of helping pharmaceutical companies with more than 45 BLAs, MAAs to market, commercializing in more than 80 countries and with many expedited review designations. Catalent, for formulation, fill and finish in the US, definitely one of the top aseptic manufacturing companies producing vials with isolator technology. Rovi, for formulation, fill and finish for international countries, experienced in 65 markets, including the US, and with a lot of vaccine experience. We are also finalizing agreements with other partners. I cannot thank enough our manufacturing partners. I have not seen in my career such a tremendous collaboration and purpose among employees of different companies. Next slide. So, how are we designing the product to be in the market? We will have multi-dose vials with 10 doses in each vial. 10 vials will go into a carton. Cartons will go into a case and cases into pallets. The product will be stored at -20 degrees Celsius or -4 Fahrenheit, which is the normal standard freezing temperature. Frozen food and freezers at home target the same temperature. We all are familiar with it. Next slide. We are designing the supply chain for COVID mRNA 1273 to be fully flexible. We expect the product to be stored at -20 degrees Celsius for a minimum of 6 months. As we get more real-time stability information, we may go beyond 6 months. In addition, the product can be up to 7 days in the refrigerator at +228 degrees Celsius. Again, this could be longer once we have more real-time stability. In addition, the product can be up to 1 day at room temperature during administration. All of this allows to use existing market infrastructure. Finally, the product is ready to use as is. No dilution or special handling is required. Next slide. By using existing infrastructure, there is flexibility to send different quantities to the different needed locations. Bigger quantities to large immunization centers, or for instance, a single pack to a small nursing home or doctor's office. For immunization, just draw 0.5 ml and inject. Next slide. COVID mRNA 1273 is a liquid vial. The other vaccines in our pipeline are designed to be lyophilized or freeze-dried, as you prefer to call it, which could allow for 18 months or more at refrigerated conditions, +228 degrees Celsius. For instance, our Cmv vaccine candidate is, as you can see already, in lyo form. You may be asking yourselves why we didn't go lyo for COVID-1273. Well, there is not enough lyo capacity in the world for a global pandemic of this nature to be produced in lyo. I can also tell you that we have an active technical development program intended to have a stable 228 degree liquid formulation. Next slide. Our CMC readiness is well advanced for a BLA and emergency use authorization. First, we count with a very experienced management team with an impressive track record in product development, BLA preparation and launch, and running commercial operations. As discussed before, we are privileged to have second-to-none partners to help us in our mission. Third, we have validated our first commercial scale, and the next scale is well in progress. Our manufacturing plant has produced above 100 GMP mRNA batches. And finally, we are having real-time and constructive dialogue with regulators. In the right-hand side, you can see a picture of real COVID mRNA 1273 vials intended to go to market. I have personally brought numerous products to market in my 30-year career, and I'm very confident to make this one happen too. Next slide. We are on target. We are bringing together the infrastructure to produce 500 million to 1 billion doses per year. We are already actively manufacturing for market use, and so far, our scale-up and documentation is on track to deliver. Next slide. Before I hand it over to Stephen, I want to sincerely thank employees, manufacturing partners, supplier partners, and regulators and government agencies for an incredible collaboration and tireless effort. This is indeed unprecedented. Stephen?
Thank you very much, Juan. So I'd like to take the closing few minutes of our prepared remarks today and update on a couple of activities in the new research and development space. I'll remind you that we generally do not talk about all of our preclinical research and our extensive investments there, but we have a long-standing and major strategic commitment to continue to push the boundaries of how we use our mRNA technologies and to create an expanding pipeline in all of our core and non-core therapeutic areas. But we do regularly update when we do deals. And in this case in particular, we announced 2 partnerships today that we wanted to provide a little more context on. So the first on slide 158 is a new partnership with Vertex, expanding on our multi-year collaboration with them in the field of cystic fibrosis. This new announcement that was made yesterday is aimed at expanding into gene editing and gene therapy technologies as an alternative approach to treatment of cystic fibrosis. And I'll provide in just a minute a little more context of how these 2 different approaches to addressing this disease will operate in parallel and the differences in how we approach them. Initially, we're going to be focusing on continuing to use the novel lipid nanoparticles and mRNAs that we've been developing with Vertex for the first program, but expanding those into the gene therapy space. And importantly, benefiting directly from Vertex's ability to bring the substantial number of gene editing therapies and technologies that they've acquired or built internally over the last number of years, and bringing those payloads into our existing delivery infrastructure and platform technologies. Moderna is going to be leading the efforts around discovering and optimizing the mRNAs and LNPs and partnering with Vertex to develop the payloads as described. There's a $75 million upfront, as Stefan described, and appropriate milestones and tiered royalties as we move forward. Now, just a little bit on slide 159 to contrast these 2 efforts for those who are perhaps asking, how do we do 2 deals in cystic fibrosis with Vertex? So first, in the center, there are approximately 10% of people who suffer from cystic fibrosis who are not going to be addressable with the CFTR modulators that Vertex has been pioneering and leading that have been transformational in the treatment of the disease. Unfortunately, for those with those genetic predispositions, alternative approaches are necessary. And several years ago, we announced the partnership on the left, which will be continuing and is continuing to show progress as we announced a couple of months ago. And that is delivering a messenger RNA that itself directly encodes for the CFTR protein that is missing or not functional in people who suffer from cystic fibrosis. That CFTR protein then can normalize chloride ion transport and address the disease. We've been working incredibly hard with Vertex to advance a candidate for CFTR based on mRNA delivery of CFTR protein and providing updates in the near term, but as we have announced over the past year a couple of times, we've been making good progress towards that objective. The deal announced yesterday, the extended collaboration now in gene therapy, is described on the right here. In this case, we'll be using the same or other delivery technologies and encoding for a gene editing or gene therapy approach. Now, in the example in the cartoon on the right, we've shown a Cas9 protein, a CRISPR protein that can be used to directly edit our DNA. But it's illustrative of many different payloads that Vertex, through their extensive work in the gene editing gene therapy space, can bring to bear in this partnership. We will deliver that mRNA encoding for that protein as well as other payloads and seek to directly edit or otherwise replace or modify the gene for CFTR inside the lungs of people suffering from the disease. And therefore, instead of just expressing the one protein for CFTR, hopefully lead to a durable addressing of their clinical syndrome. Both approaches are moving forward, and we're excited to continue to expand our commitment to cystic fibrosis and work with Vertex, who is a unquestioned leader in both addressing the unmet need in cystic fibrosis, but increasingly a leader in the gene therapy space as well. Now, in slide 160, we did announce another partnership as well yesterday with, in this one, also dealing with a pulmonary disorder, with Chiesi. The Chiesi Group is a rare disease Italian company that many of you are probably familiar with a strong commitment to innovation against rare diseases, including pulmonary rare diseases. And in particular, this program is aimed at the discovery and development of mRNA therapeutics against pulmonary arterial hypertension, a rare disease that infects 2 to 5 people per million adults. It's a progressive and terrible disorder resulting in high blood pressure in the arteries of the lung and ultimately right-sided heart failure. there is substantial unmet need, in particular for novel treatments that could delay or reverse the progression of the disease in patients. Now, this is our first partnership with Chiesi, and we found a great match in our commitment to addressing rare diseases and to using innovation to get there. This partnership, under the partnership, Moderna will lead the discovery efforts, and Chiesi will be leading the development and commercialization worldwide, but it will really be a true partnership throughout. Moderna will receive a $25 million upfront and is eligible for development, regulatory, and commercial milestones, as well as royalties based on the progress of the programs we hope to move forward together. And with that, I'll turn it back over to Stefan for his concluding remarks.
Thanks, Stephen. Before closing, I would like to thank our teams and our guests for their presentations today. And I would like especially to thank the Moderna team, our partners, our suppliers, all our collaborators for the incredible last 9 months that everybody has been working so hard trying to stop this pandemic. mRNA could be a new class of medicines. That was our vision. This is how far we have come. As Juan told you, those are actual photos of 10-dose vials that are ready to be used and shipped if 1273 is approved. The team is working hard to make as many products as we can while ensuring high quality and respecting, of course, all the rules of making GMP products. On the right, you have a mock-up of the cardboard that the team actually has been working on the rural version and be ready for shipping. If you go to 164, Moderna has and will continue to expand in 2 dimensions. That has been our vision and our strategy since the beginning, and we're not slowing down. The first dimension on the x-axis is time, which is going from the concept of a medicine to the commercialization of a medicines. Go from an idea of a drug, in preclinical research, preclinical development, phase 1, phase 2, phase 3, and commercial. This is very typical to pharmaceutical and biotech companies. But the other dimension in which Moderna is expanding regularly, and we are very committed to keep expanding to maximize the potential impact that we can have on patients, is around the breadth of mRNA technology. We started to win a clinic with vaccines, oncology, VEGF injected directly in mRNA into people's heart, rare disease, the work that Stephen and his team are doing with Vertex team on figure out how to deliver safely mRNA into the lung. And with last night's announcement on gene editing, we just want to continue to expand, exploring how we can help as many patients as we can by expanding the use of our technology. And on slide 161, you see basically the current pictogram of where we are as a pipeline, as a company, trying to continue to advance the pipeline to the right to launch those important medicines and continue to advance by adding new breadth to the company. Slide 166 is a usual slide of company update and profile, COVID-19 in phase 3, CMV, PCV, OX40 ligand 2 program in phase 2 in cancer, and VEGF in cardiovascular, 8 program in phase 1, now 12 positive phase 1 clinical readouts. The portfolio, is very strong and very diverse. 7 vaccines for major unmet needs, 5 immuno-oncology drugs that are all in clinical studies, all combined with commercial checkpoints, 4 rare disease program, 2 autoimmune disease program, 27 healthy volunteers and patients enrolled, more than 1,100 employees, very strong manufacturing infrastructure, partners, and a strong cash balance. As we close, let me share a few thoughts. Moderna could receive an EUA for mRNA 1273 in 2020. Moderna could receive a BLA approval for mRNA 1273 in 2021. mRNA 1273 could validate the entire vaccine portfolio because mRNA is an information molecule. The phase 3 success in mRNA 1273 could have implications for all the vaccines in development, other vaccine in the research pipeline, other vaccine that we are not yet working on in the labs. That is the power of an information molecule. I believe 1273 could transform Moderna and create a unique and historic inflection point for the company. With a manufacturing base plan of 500 million doses per year that Juan and his team are working to potentially get up to 1 billion doses per year, Moderna could have a multi-billion-dollar revenue line in 2021. That will create a very strong balance sheet for the end of 2021 because we have $1.3 billion of cash as of June, and the multi-billion dollar of potential revenues will, of course, add to that balance sheet. If we can execute and achieve that, that will allow Moderna to invest in R&D, to scale the development pipeline, and maximize what we can do for patients. Our strategic priorities are very clear. One priority is to maximize the impact of 1273 access and thus value creation of this product between now and the end of 2021. This will allow us, priority number 2, to accelerate vaccine development, to accelerate as much as we can product in the pipeline, to bring new vaccines to development like flu as we discussed today, and many more. It will allow priority number 3 to generate multiple human proof of concept in oncology, rare disease, and autoimmune diseases. And this will allow priority number 4 to continue to invest to expand the breadth of mRNA and how we can help patients by investing in mRNA technology improvements, delivery technology, long delivery, gene editing, and Stephen and the team have many, many more ideas. I believe this is a historic moment for the company. I'm extremely thankful for what the teams have done to get us to this point, and we are extremely thankful to our long-term investors that have provided over the years the capital so that we can build the best version of Moderna that we can. With this, thank you for your attention. We'll now be very happy, operator, to move into Q&A.
Thank you. To ask a question, you'll need to press *1 on the telephone. To withdraw your question, press the # key. Again, if you would like to ask a question, press *1. Our first question comes from Matthew Harrison with Morgan Stanley. Your line is open.
Hi, team. This is Max Scoron for Matthew Harrison. Thank you for taking our questions. To start off with, can you comment on the potential read-through of the repeat dosing data to the rest of the pipeline, specifically LNP clearance and pharmacology? Also, are you measuring the functionality of the antibody? Finally, can you extrapolate anything from the CMV data to the COVID-19 vaccine? What are your thoughts around potential durability and the portion of induced neutralizing antibodies that are functional? Thank you.
So this is Paul. Thanks, Max, for those questions. You asked me a few. I just want to make sure I don't miss any, so if I do, remind me. Read through on the platform. Look, I think the repeat dose is clear on two really, or three critical fronts. Number one, when you do see adverse events, they're mild, they're gone within a day or two, and there's no accumulation of any adversity. And I think that's a critical point to establish. We had assumed that because of what we saw in the single dose, but it's very reassuring to see. You come back again, you get the exact same profile, either nothing or grade 1, grade 2, relatively minor and transient effect. Number two is the translation, the pharmacology. Do you repeat to the same extent you had? And I think the answer here is yes, and it's pretty clear. Number three is you look at the PK curves and sort of the lipid nanoparticle, and it's pretty clear that clearance is rapid. Clearance is much more rapid than the protein, which is what you would expect. That's the beauty of this technology. The mRNA gets in, the LNP is rapidly degraded to all of its components, and then they're gone because we've engineered it to have a rapid short half-life. What you get is the stackable pharmacology. And so I think that really bodes well. And the final point I'd say is look at the doses at which we're seeing potentially therapeutic levels of protein at 0.1, 0.3, 0.3 given repeatedly. That's the dose range at which we anticipate to have benefit for the children. The second question you had was on durability, I think, in the read-through between CMV. Oh, I'm sorry, you also asked about the activity of that protein. Yes, it is active. It is active because we measure its activity based on neutralizing. We had demonstrated that in the past. I didn't bore you again with the data, but the protein that we make is indeed functional because you can demonstrate neutralizing activity of that monoclonal antibody that gets formed. To your question on durability, and CMV read-through, I think that you can see with CMV the unique ability of this platform to boost. And why do I say that? Because if you fast forward to COVID, we see some platforms like ours with mRNA that can boost repeatedly, and I think the CMV data are proof of that, versus other platforms, namely engineered recombinant viruses, the adenovectors for one, where I think boosting is likely to be more challenging because a lot of the immune response is against the vector as opposed to the transgene. In the case of mRNA, all we are doing is just showing the body that singular protein. The beauty of the CMV data is that you can see that, again, in functional readouts for neutralizing activity against two separate antigens that we can continue to boost. You've seen the confidence around selecting a dose for phase 3. I think that bodes well for durability on COVID. I think the last point to sort of extrapolate, I think it's early days to know how and what is a durable immune response for COVID. I think my sense is that people who've gotten sick do not get sick a second time. Therefore, for the duration that we understand this virus, at least 6 months, the durability is there for infection. And since we can elicit the same level of antibodies or higher, then we anticipate to have durability at least as good as from wild-type infection. I do think that if you put it all together, we see the potential down the road to also use our technology to boost the immune response further against COVID just to make sure that people remain protected. Over.
Great. Thank you.
Thank you. Our next question comes from Ted Tenthoff with Piper Sandler. Your line is open.
Hey, thank you very much, and thank you for all this data. Really great to see the progress, not just on 1273, but on the pipeline more broadly. And I just have to say, amazing what the difference a year will make, not just at the company, but in the world as well. I wanted to dig in a little bit more on the concept of adding the flu vaccine and even the potential for flu plus COVID-19. How would you envision, and appreciating your kind of just announcing that, but how would you envision developing that? Would this be something where you would have to advance the flu vaccine itself and then ultimately combine them? Or would the whole goal be to just do flu with COVID-19 for seasonal? Thanks.
So that's a great question. Oh, Stephen, go ahead.
No, you go ahead, Tal, please.
Well, you can tell we love answering this question because we've been thinking about it a lot internally. I think the beauty of our platform for flu is that we envision the ability to react quickly, right? And so one of the challenges with flu is that to match this year's need with a vaccine, and I think a technology that can react so quickly, and you've seen us do that in the case of COVID, I think that bodes well for our ability to provide a unique solution for flu. Now, of course, the challenge is that with flu, we're going to need a different vaccine every year. And the question is, what will happen with COVID? And I think it's early days. So there's two scenarios. One is it's the same COVID, but people want to have a boost or people need to have a boost. And we'll see that over time, in which case it's pretty straightforward to combine our vaccine, the one for COVID, with whatever annual variant you have because of the ease of combining mRNAs in the same vaccine, and I think we've now demonstrated that ample times. The second possibility is that actually you will start to see immunological escape mutations for COVID arise. Now, that's still not shown to be clinically relevant. People have started to describe it, but it's unclear whether that's going to be of epidemiological significance. If it is, then we would simply do the same, and we would make sure that we move quickly on both. Not having to have an egg-based sort of chicken farm platform, but rather one that is digitally encoded and can move rapidly, I think is going to be an advantage to our ability to serve the need of in either scenarios. Over.
Yeah, and maybe let me just add one piece, which I think goes back to a medical need, which is if the flu market was well served with high efficacy vaccine, we will not bother. Because as you know, we like to go after big medical problem, high unmet medical need. This is where we want to apply the platform, given all the things that we could do in theory with mRNA technology. And I think the elderly data that we got, just push us in that direction even more. As you know, even other mRNA technologies have shown a drop from young adult to the elderly with COVID. We believe that all the investments we made in science over the years, formulation, manufacturing process, explain why the elderly data is so strong. So between what Tyler was saying around the strain, the mutation, the ability to have high efficacy, if we could have a high effective flu vaccine that we could then potentially combine, as Tyler described, we could have a big impact on reducing death, on reducing hospitalization, and on reducing the time people waste by being even sick when they are likely sick and miss work and so on. So we think it's a big medical problem. We think we have a chance to make a dent on it, and we're going to go after it.
Very, very helpful. I appreciate it.
Thank you. And our next question comes from Salveen Richter with Goldman Sachs. Your line is open.
Good morning. Thanks for taking my questions. Two for me. So can you help us understand your assumptions that get you to a November base case for the first interim look for the COVID-19 vaccine and then an EUA by year-end 20, given the probabilities that you mentioned today? And then secondly, could you just provide your thoughts around rare side effects associated with vaccines, particularly given the neurological safety events we saw with Astra, including transverse myelitis? Thank you.
Hi, Salveen. Thanks for the questions. It's Tal. Assumptions for the November base case. Well, I think that there is a likelihood that we will reach 53 cases in November. Now, so if the vaccine is highly effective, in the 80 to 90% range, as Jackie walked you through the math, then it's a question of getting lucky that we actually have the split in the sample size that we have, because admittedly 53 is a relatively small number. But if the vaccine is very effective, then there is a good likelihood that we would cross that barrier even at 53. And if that's the case, then I think that's a scenario where we have data in November to support an EUA. I think FDA in their dialogue, I'd say two things. One, there's a very strong collaborative work between the agency and I think all companies, certainly ours, so that they're aware where we are and we're working hand in hand to ensure that it doesn't take them long to approve an EUA once we cross the threshold and they have the ability to query and get comfortable with our data. So I think that's the scenario here. In terms of your second question on rare side effects, so a couple of points. Of course, the transverse myelitis that's been described, I think it's early to say to what degree it's related, but we all want to be conservative. The scientific hypotheses that I've seen and sort of make sense to me have probably more to do with an adenovector platform than they do with the antigen per se or anti-spike protein immunity. I think the second point is that we've not seen a similar case, nor have so far have we been asked to change anything in terms of the conduct on our trial. And I think the third point is I'm reasonably confident that if it were to occur, we would have seen it. In other words, we have very robust safety monitoring embedded in this phase 3 protocol. It's not just us, but it's our NIH collaborators, and any question on any case or any adverse event that people are worried about gets escalated quickly to the Independent Data Safety Monitoring Board. So, it's always something you look out for vaccines, and you will never hear me say that our vaccine is safe, because safety is always, defined over time, and it's related to what we're trying to achieve. So we never rest on the laurels of feeling good about it, in the sense that we always continue to collect the safety data. Having said that, so far so good for where we are in terms of our vaccine. Over.
And maybe to just add a little bit to Tal, and maybe Juan, you might want to add a bit of color. The other piece that's important, Salvin, to enable a potential EUA in that time frame is the CMC work that is ongoing, both within the company, where the teams have been working really hard to get us kind of BLA ready, so that CMC is not in a critical path. That is really important. And there is a very close dialogue with the agency on the CMC side, so that everything is done before the clinical data comes out, so that the clinical data is really the bottleneck to a potential EUA, that we don't then, need to have, meetings and data and other pieces of the EUA missing. So there's a lot of work going on there that Juan and the team are leading to make sure that CMC is not critical path. I don't know, Juan, if you want to add any color.
Yeah. So, for instance, one of the strategies that we have followed is, as we scaled up, as soon as we completed the next cycle of scaling up, we have validated the process. So we have put together the PPQ packages so that we are ready almost at every moment as we go forward. And obviously, these packages, this information is being shared with agencies as in real time in a very transparent way.
Thank you.
Thank you.
Thank you. And we have a question from Michael Yee with Jefferies. Your line is open.
Thanks. Appreciate all the details and programs today. Platform question relates to the fact that the difference between this year and last year is that you've dosed 25,000 people now for COVID, and presumably could be millions of people over the next year or two. How should we think about how much de-risking of the platform or the LNP or mRNA specifically could be extrapolated to different parts of your other programs, whether that be oncology or more likely prophylactic vaccines, which I think is most de-risked? So just maybe make a comment about how much will have changed in the next year after dosing all these people. And then the second question relates to information yesterday from Lilly suggesting there was 6 to 8% resistance virus, at least to that antibody. Maybe just make a comment about whether that surprises you with the fitness of what's going on with COVID, or any implications as it relates to the development of the vaccine. Thank you.
Thanks, Michael. This is Tal. Let me take those. I absolutely agree with you that I think the increase in the safety database that we've experienced so far, and we've gone from hundreds to thousands to tens of thousands, and hopefully, knock on wood, in the not too distant future, we'll be able to bring benefit to millions. That, I think, provides tremendous reassurance for everybody regarding the fundamental of mRNA vaccines, and I do think there's a read-through to the mRNA platforms. Admittedly, therapeutics are higher doses given repeatedly, but so far what you've seen is, if you follow the science, what you see is what you expect. In that regard, we've been, and I think everybody has been reassured, although I'll say, we've not been surprised. This is what we've expected based on the scientific principles that underlie what it is we're trying to do here. mRNA as a molecule is nothing new to humanity, nor is it new to evolution. It's one of the oldest forms of molecules known to man. To your point about the Lilly and resistance, I think there's a big difference if you're talking about a single monoclonal antibody as something that generates selection pressure during the time of an ongoing active infection and viral replication in somebody's body, versus a very polyclonal response elicited by a vaccine ahead of an infection. People have described resistant clones come up to this antibody or that. They've not yet translated into an epidemiologically relevant escape. I think if we did that, people would probably start calling it SARS-CoV-3. I think it's why Regeneron, for example, has gone from the get-go with a combination of two antibodies, and now if they think just two will be enough to get over the problem, then certainly when you have thousands, which is what I would expect the case for a vaccine, and certainly we generate a very nice broad response. Part of it is against the receptor binding domain, but clearly we have epitopes outside that, and even within that, you've got a polyclonal response. So I don't think that that's relevant, on first-order scientific principles to expectations of a utility for a vaccine. Over.
Thank you.
Thank you. Our next question comes from Gina Wang with Barclays. Your line is open.
Thank you for taking my questions. The first set of questions is regarding the phase 3 trial design. Just wondering, first, is there any fertility embedded in the interim analysis? And also regarding the trial size, I think Pfizer increased the trial size. Do you feel comfortable or confident that 30,000 patients will be sufficient to reflect, different population? And lastly, 60% efficacy, when you mentioned that, just want to confirm that means 60% reduction of positive COVID-19 versus placebo. And I have a quick manufacturing question. Is just wondering, have you already started the commercial doses, and how would you split in the future between U.S. and the EU?
Thank you, Gina. There were several questions embedded there. Let me take the first few on trial design, and then I'll defer to Stefan. Yes, we do. There is a look for futility. It's sort of the foremost statistical look. It's handled by the independent statistician within the Data Safety Monitoring Board, because we want to make sure that things are going the right way, not the wrong way. In terms of trial size and relevant populations, if you look at our performance on the diversity, I think, and we've been very transparent with it, what you've seen is over the past few weeks, we have continuously improved our ability to reach minority populations and ensure that the doors open wide and long for them to have the opportunity to enroll. So I expect that at the end of the day, 30,000 subjects should give us a pretty good representation across the different risk strata, be it age, minority, or comorbid conditions. Now, the truth is that confidence is much less a function of the trial size, and it's much more a function of how many events will you see at the time when you decide there's, or it's apparent that there's efficacy. Because obviously, if that efficacy is driven by a very small number of cases, then it doesn't matter whether you've got 30,000 or 100,000. It's all resting on a few dozen cases that make that determination, and that's an interplay, frankly, between how effective the vaccine is, as Jackie has described it, and the statistical rigor and analysis, and the wish we all have to make sure that we don't unduly delay an effective vaccine for the population. The last question you had was, yes, 60%. The calculation is done simply for vaccine efficacy. It's derived by dividing the rate that you get within the placebo versus, within the vaccinated versus the controls, and that's the reduction in the risk. Let me hand it over to Stefan.
I think you answered everything perfectly, Tal. Did he, Gina? Anything else, Gina?
I think there was a question on the commercial, on how we intend to distribute between U.S. and ex-U.S., if I recall.
Yeah, so on the U.S., as you know, we have an agreement with the U.S. government for 100 million doses. And so our intent is to work very closely with Operation Warp Speed, with the CDC, with the U.S. military to get the vaccine to central nodes of distribution. As you have seen, McKesson has been retained by the U.S. government to do the physical distribution. And so that's kind of how it's going to operate. The allocation in terms of population at risk, in terms of different geography and states, will be decided by the government, not by Moderna. And outside the U.S., it will be done through the outside the U.S. supply chain that Juan and his team have established. Lonza for making formulated product, Rovi in Spain for filling the vials. And that would be the same thing, I would say, shipped directly to governments that have placed orders already or the orders to come as we continue to engage with government and to figure out how we can be helpful. And this is how it's going to be done. Does that make sense?
Yes. Thank you.
Thank you, Gina.
Thank you. We have a question from Cory Kasimov with J.P. Morgan. Your line is open.
Hey, good morning, guys. Thanks for taking my questions and really appreciate all of your transparency around this phase 3 program. So speaking of the phase 3, I'm curious how we should be thinking about infection rates in the COVE study relative to the reported SARS-CoV-2 infection rates. I get that you've selected site locations that are trying to be near COVID-19 hotspots, but interested to get your view as to whether the confirmed case rates are underreported or if there's a specific strategies to enrich your trial for events. And then my second question is, I'm wondering if you can speak to any assumptions around the potential timing of the second interim, if it gets that far, given the November commentary for the first plus the lower statistical bar you have for the second one. Thanks a lot.
Hi, Corey. It's Tal. Let me take those. Let me jump to the end game. I think it's too early for us to say whether the infection rates are exactly what we would anticipate based on the zip codes where the trials are accruing. What we have done in the trial is try to ensure that we have a representation of the risk factor. And by the way, to enroll, everybody needs to be at higher than the usual risk. That is, they have to have a risk factor, whether it's by their occupation, that they're out there, they're not like me sitting at the computer all day long, they're elderly, they have comorbid conditions, or they belong to minority communities. And so with that, we've tried to enrich for the relative expectation of cases. On the flip side, often clinical trial participation doesn't necessarily one-to-one match the demographics of any given zip code. So we've taken active strategies at every local level to try to ensure that there's open likely to get infected and most likely to get infected. But it's too early to say whether the rates are actually on par with expectations. So we have a pretty wide range of when the cases could happen. In terms of your question on the timing of the secondary analysis, I think it's important to note that it's not a linear. It's not going to take the same time that it took from the start of the trial to get to 53 as it is going to go from 53 to 104. I think going from 53 to 104 will happen much quicker because you're now sort of at the linear part of the curve. Everybody's already been enrolled. Everybody's being followed up. So you're constantly accumulating follow-up, and it's a function of the follow-up time that you expect to see cases. So I expect the delta between those two to be a matter of weeks, maybe a couple of months, probably not longer, based on current expectations. But again, all of this is subject to varying predictions. We look and we try to project ahead, and I can tell you that every week when we look ahead, the variance in projections is probably in the order of 2 weeks of what to expect, sometimes earlier, sometimes later. So I think our ability to project the movement of this pandemic is too imprecise to give any more, I think, precision on when we would expect those cases to happen. Over.
Okay, great. Thanks a lot, and good luck with the progress.
So thank you so much. Before the next question, I just wanted to clarify one point that was in the previous question, which referred to lower statistical criteria at subsequent analyses. I wanted to be clear that actually the statistical criteria are the same throughout the trial. So these criteria are aligned with the guidance that FDA issued earlier in the summer, and they align with a lower bound of 30%. The difference between each subsequent analysis is that we have a higher sample size of cases, and with a higher sample size, you have a closer estimate of the efficacy through your 95% confidence interval. So that's really why the likelihood of meeting your criteria increases given the same vaccine efficacy assumption.
Thank you, Jackie.
Our next question comes from Jeff Meacham with Bank of America. Your line is open.
Hey guys, this is Alec on for Jeff. Thanks for taking our questions. Just two from me. So first on the chikungunya antibody program, could you just remind us as to the benefit of expressing an antibody with mRNA rather than just administering the antibody on its own, especially given what it looks like as the need for frequent repeat dosing? Is it mostly the dosing interval, or are you also looking at the antibody quality as well? And then my second question is on the seasonal flu program. How was your clinical experience with 1851 for H7 and 9 and 1440 for H10 and 8 influence the way you're thinking about the continued development for flu, including the use of endpoints such as the hemagglutination inhibition titers? And would you seek to partner the flu vaccine programs out with, say, the NIH or other parties, or are you really looking to take it forward on your own? Thanks.
So, Alec, let me start. The benefit for us, well, first of all, this was the first opportunity to demonstrate protein functionality. I think monoclonal antibodies are probably the most widely used and best understood of protein types of drugs. So I think it's a great proof of concept. More to the point, you can't make the wrong antibody with an mRNA technology, right? So I think the veracity of the biology that you translate, and once you make the antibody, you know that antibody is going to be functional. And I think that is a potential advantage of this technology that has to do with all the efforts you need to do in the recombinant space that here the body takes care of it. I do think there's a world ahead where the potency, rapidity of actually making something new and potentially even cost of goods would make this platform a beneficial, even in the monoclonal antibody space. It's not immediate, it's not necessarily today, but I think that future is there. The final piece is that there's an opportunity to do combinations that for us on a relative cost of goods basis would be a very significant difference than what it does in the monoclonal antibody space. There are a few examples of antibodies combinations, and they usually max out at 2 for obvious practical reasons. I think we see a future where we could leverage the power of this platform to do combination mRNAs to go even beyond that into a polyclonal or a polyepitope or even a polydrug type of a product that would be based on monoclonal antibody recognition. Seasonal flu. Let me take a first stab at that. I think we've always been excited about the potential we have for flu, because if you go back to the H7 data, the rate of seroconversion that we saw was practically in everybody. And that's been a reassuring feature of our vaccines. It's true of COVID. Look at the CMV data. The error bars are actually quite small. When somebody gets immunized with our platform, they mount an immune response, and it's pretty clear, and it's robust, and it's neutralizing. And so we had seen that from the early days. In fact, there's no other published reports that I'm aware of H10, but for H7 there were. And proteins alone didn't do much. Proteins with an adjuvant sometimes got there halfway, but we can consistently get there, and we can do so with a really rapid turnaround time. I think the advantage we have now is having demonstrated combinations, and now that we will have set up a manufacturing infrastructure, I think that all puts us in a well-poised position to enter that space, as Stefan had said, and I'll defer to him for any additional comments. Over.
Yeah, and thank you, Tal. Maybe just one thing on the seasonal flu to add to Tal's remark is that do not forget, if you have a curiosity, to go back after today's announcement and look at the human data and preclinical data, but especially human data published in vaccines for both H10 and H7 in the same paper. Remember, this was what Stephen will call kind of dark ages Moderna technology version. This was with an old standard lipid that was not biodegradable, not the lipid we use now in Cmv or 1273. This was done with old mRNA potency technology. This was done with very old manufacturing process. And so I will not be surprised when we go into the clinic with a seasonal flu product that will have even a better performance to what we already saw at the time. As Tal said, most of the participants, if not all, were with a HA titer that was above the FDA approvable endpoint, which we use as our bar for assessing success of this program. So I think this is going to be quite interesting going back to the clinic with seasonal flu with the newest version of the technology and all the investments the team have done.
Great. Thanks a lot.
Thanks, Alex.
We have a question from George Farmer with BMO Capital Markets. Your line is open.
Hi, thanks for taking my questions. Can you talk a little bit more about the Th2 response that you're not seeing in the phase 1 study? I mean, now you're kind of referring to this as perhaps a good thing. Some of your competitors are boasting that they can elicit a Th2 response. How should we think about that as within the context of your vaccines versus others?
So this is Paul. Let me take it from a clinical perspective. I think the focus on the Th2 in this stems from the observation that when people went back to those vaccine-enhanced respiratory diseases that were seen in the case of formalin-inactivated protein vaccines or viral vaccines with RSV and measles back in the 60s, and they try to analyze the immune response in those children, it appeared that you get a more Th2 geared, and I think of Th2 broadly speaking as sort of your allergic, anti-helminthic, anti-parasite response as opposed to the Th1, which is more towards viruses and intracellular pathogens. And so with that context, people have been trying to look at the balance of Th1, Th2, with the expectation that you really are looking to get here is Th1. What we've done is a whole bunch of, we and our collaborators, is a lot of correlative preclinical work primarily geared around the safety question, looking at aged mice as one extreme, and then looking at every species, evaluating histopathology to reassure ourselves that indeed when we're preventing infection or preventing disease, there's no exacerbation anywhere. And that continues to correlate. Now, it's a correlate, it's not a causative, but it does continue to correlate with the Th1 response as opposed to a Th2 response. And so when you put that all together, seeing a Th1 response for our platform consistently and correlates of protection, and then we see it in the clinic, I think that's really reassuring. As I said, I think it would be worrisome, especially if I had a protein or an adjuvanted protein to the unit vaccine, and I saw more of an allergic type of an immunological profile. It's not proof and it's not causative, but that's the reason people look at these profiles to begin with, because of those correlations back dating back decades of what was seen with RSV and measles. Over.
Mm-hmm. Okay. And then in the phase 2 trial, you mentioned that all fully enrolled, everyone's received a booster. Will we be able to get a look at the neutralizing antibody responses, whether using the print assay, the neutralize or the pseudovirus assay, in the near term, rather than having to wait for the full 13 months of follow-up?
Well, it's not going to be 13 months, but I do think we're going to wait for the endpoint as defined, which is a couple of months. And frankly, right now, the important thing for me was in that trial was to get a sense of reassurance of safety on the way to phase 3, and then over time to understand how 50 performs relative to 100. So we will share them when we have them. In terms of decision making, stay tuned, I guess, and once we're able to show them, we will.
Okay, great. And then one final question on the Cmv vaccine. So how should we think about the market for this vaccine? Is there a market, in fact, in patients or in individuals who are seropositive, or should we just think about potential penetration in the seronegative market?
So the expectation here is that this will be utilized irrespective of serostatus, because if you had to start to test for people, it would actually end up being a real barrier to access. And so if you talk even to the FDA, certainly the folks that we've spoken to back at the CDC, there is an expectation that this will be for everybody. And the FDA was clear in stating that they don't expect us to demonstrate benefit for the seropositives, only safety, but they do expect us to demonstrate safety for seropositives because of the expectation of a label that will be inclusive for everybody. I will say this. I'm super curious as to whether the ability to boost even seropositives by the amounts that we are, remember these are people who are living every day with the virus, and there is some homeostasis between their immune system and a latent virus infection. I'm super curious to see whether the effect that we can focus the attention of the immune system on these antigens to the level that we sort of reset the homeostasis for these people may have a benefit as well down the road. But admittedly, it's going to be really, really challenging to demonstrate that benefit, and I don't expect us to be able to do that pre-licensure, hopefully post-licensure.
Mm-hmm. Great. Okay, thanks very much, Tal.
Our next question comes from Mani Foroohar with SVB Leerink. Your line is open.
Hey guys, thanks for taking the question. First of all, I'd like to echo Corey's sentiment regarding congratulating you guys on showing tremendous transparency, and hopefully some of your competitors in the U.S. and elsewhere will follow suit in that regard. In terms of questions, a couple quick ones. First of all, for you, Stephen, for the partnership with Vertex, is it reasonable to assume that both sides of the partnership are pursuing an inhaled approach to delivery for CF? For Tal, regarding 1273, you commented, and I may have misheard it because you cut out there for a second, that you expected the 53 events perhaps in a November time frame, which sort of explains that discussion around November. Did I hear that right? And should you not hit the stopping threshold for efficacy at those 53 events, is it reasonable to expect the 106 and 151 events closer to, say, a December-January timeline based on average event rates you're seeing right now in the study? And then as a third question, one of the nuances around the COVID-19 disease itself is that it appears to be it tends to be severe more frequently in men than in women. Do you see a biological component of that based on the data you've seen in your early studies and your study of the virus preclinically internally, or do you think that's more driven by behavioral and cultural factors between the genders in the places where the majority of COVID-19 burden is being seen?
Sure. So
let me maybe take the first.
Yeah, I'll take
Stephen, you want to go first?
Yeah.
Thanks.
So just very narrowly on the question on the Vertex partnerships, we haven't disclosed, but the short answer is we are looking at all sorts of delivery, including inhaled delivery. And we obviously believe the fastest way to access the lungs is going to be through an inhaled route. And so both of our partnerships with Vertex do look at pulmonary delivery through the inhaled route.
Thanks. And on your question on events, yes, I think there is an expectation for 53 events to occur in November. Sometime in November is sort of a reasonable base case. And then the next analysis would take place at 106. The likelihood, I think Jackie walked through how the likelihood is a function really of the number of cases as well as the actual vaccine efficacy at any given point. The question around more severe in men, I don't think I've got any wisdom to add here to what's out there in the field. It is plausible that there are biological factors that relate to gender here. I don't think we've not seen any differential effect in any of our trials in terms of vaccine performance or antibody levels. And so I don't necessarily expect to see a gender difference in the performance in terms of preventing cases. Whether those reasons have to do with behavior and therefore infectious load to begin with, which seems to be one plausible mechanism for explaining why some people get more sick than others, or whether they have to do with inherent biology, I don't think anybody knows at this point. Certainly I don't. Over.
Great. Thanks a lot. That's really helpful. And congratulations again on all the progress.
Thank you.
Thank you, and I'm showing no other questions in the queue.
Great. So thank you so much, everybody, for joining us today. The slides, of course, are available on our website. Stay safe, everybody, and speak to all of you very soon. Bye-bye.
Ladies and gentlemen, this concludes today's conference call. Thank you for participating. You may now disconnect, everyone. Have a